and continue on with chapter 16 here, special senses. All right, so where we picked off, or left off, I should say, is going to be gustation, your sense of taste. Give me 1 second here, folks. I'm having a problem. There we go. Okay. All right. Sense of taste, gustation. So if you kind of understand, or if you do understand smell, then gustation is very similar, except we're just going to be dealing with the mouth instead of the nose here. All right, how we saw when we were talking about the molecules that will stimulate the chemoreceptors there in the nose, we call those odorants. Yeah, I understand. And that hopefully after today's class, Morgan, you'll have a better understanding of that. So that's not uncommon for people to have some confusion there. But when we're looking here at taste, and again, it's interesting that you mentioned that because taste and smell are, they're intertwined. And if you notice that sometimes your sense of taste is diminished when you have a stuffy nose, like with a cold or allergies, there is going to be a tie-in between those two senses. So there are a lot of similarities, which to me makes it a little bit easier for folks to kind of understand and learn this material because of the similarities. So again, whereas with olfaction, our sense of smell, we talked about the detection of odorants, those molecules that get embedded into the mucous membrane there. Here, when we talk about gustation, we're talking about detection of tastants. The tastants are going to be those chemicals that are going to stimulate the chemoreceptors, which are located in the taste buds. So let's find out where those taste buds are. And some of you might think, oh, I know, where my taste buds are. I have these bumps on my tongue. They're called papillae, and those are my taste buds. Well, that's true. Right? There are bumps on your tongue, the papillae there, right? But not all of those papillae contain taste buds. All right, so you have this, there's different groups of papillae. We're going to run through them. All right, the first one here listed on our slide here are the Filiform Papillae. And these actually have no role in taste, none whatsoever. But the job of these papillae is to help when you are chewing food to kind of give you more grip when your tongue is manipulating the food in your mouth, pushing it against your molars or whatnot. Right, so the Filiform Papillae, right, do not offer any role in gustation, but they will help with the handling of food. And we're going to find those on the anterior 2/3 of your tongue. The next group of papillae are called the Fungi Form, and we call them that because they look mushroom-shaped. And so we will find some taste buds here. And those are going to be located on the tip of your tongue and on the sides of the tongue. Our third group of taste buds are going to be our Folate Papillae. Right? The Folate Papillae, again, folia, think of autumn, fall, right now, that season that we're going through. Well, these papillae have these ridge-like configuration that look like leaves. Now, the thing with these papillae is, right, the taste buds that exist there are going to be present during early childhood. Right? Again, we don't have a lot. So these are going to be located on the side of your tongue, but more posterior towards the base of the tongue. Then finally, our last group of papillae, the Valet or the Circumvale Papillae, these are going to have, right, when you look at your tongue, if you're standing in the mirror and sticking out your tongue as far out of your mouth as you can, if you look back towards the base of your tongue, you can see these papillae there. And they're actually in a V configuration. So that's an easy way to remember because they're in a configuration like this on the posterior portion of your tongue. And so I think of that configuration, V for Valet. This is where we're going to find the majority of our taste buds. And we'll see them back there on the dorsal portion of the tongue, right here. Here are the Valet Papillae right there. And those papillae contain, all right, the majority of the taste buds for your tongue. So you'll see up here in the front, right, we'll have the Filiform Papillae. They're located all throughout in the anterior 2/3 of the tongue. All right, we'll see those also on in the tip here. In addition to the Filiform Papillae, we'll have our Fungi Form Papillae. These will have a few taste buds, and you can see they're kind of interspersed here in the papillae here. And then back towards the back of our tongue and the lateral portion of the tongue, we have our Folate Papillae back here. These are the ones that are present during early childhood. And then right here, these are going to be the Valet or Circumvale. And that's what you see here in this picture. All right, so the Circumvale or the Valet Papillae have all these taste buds here on either side. As we zoom in, right, you can see a similar type of comparison between the olfactory sense and the gustatory sense, in that you have a cell that is going to be responsible for the taste sensation, like we had with the olfactory receptor cells in the nose. They were responsible for the smell sensation. So these cells we call the gustatory cells. And then interspersed, we have our supporting cell. Right? And they, again, help to give physical support to our taste bud structure. And then we'll have the baby cells. Those are our basal cells. And so they'll replace the gustatory cells. Now, if you recall, in the nose for olfaction, the olfactory cells were replaced every 40 to 60 days. All right, and some of you may think, well, that's not very long. Well, guess what? Your taste bud cells, the gustatory cells, only last 7 to 9 days roughly. And so they'll get replaced even sooner. And at the very tip of these cells here, it's tough to see, right? We have these microvili, and the microvili are going to house the chemoreceptors. And they all come up. If you look at the structure, this kind of looks, the taste bud kind of looks like an onion. And so at the one end here where it tapers up, right, we have the microvili on our gustatory cells, and they hang out in this little space here called the taste pore. So when you eat or drink, right, whatever gets mixed up with your saliva, it's going to wash across the taste pore. And that will stimulate the chemoreceptors here in the gustatory cell. And we'll go through all that, how that all works. All right, so keep that in mind when we're talking about all those different types of cells. Very similar to what we saw, right, in our olfactory sense there, how we had our olfactory receptor cells, the supporting cells, and the basal cells. And it's pretty much the same type of scenario there. Okay, so at one end of our gustatory cell, where we are going to have our microvili, and that's going to be the actual dendritic end of that cell. And so that dendritic end will contain those chemoreceptors, which will be stimulated when the taste buds come in contact with those receptors there. All right, so how do we get our nerve signal to go from our tongue in our mouth back to our brain? So as you recall, right, with the olfactory sense, we had a sensory neuron chain. Okay, we had a primary, secondary, neurons. Well, same thing here. For our gustatory pathway, we are going to have a 3-neuron chain: a primary neuron, our secondary neuron, and our tertiary neuron. Now, the primary neuron, right, we refer to those as the sensory neurons. They are going to come, all right, from 2 different cranial nerves, depending on what part of the tongue is stimulated. All right, so let's look at the anterior 1/3 of our tongue. That part of the tongue is going to be stimulated or innervated by your cranial nerve number 7, facial nerve. The posterior 2/3 of the tongue are going to be innervated by cranial nerve number 9, glossal pharyngeal nerve. So both of those cranial nerves are going to provide the sensory neurons, which are our primary neuron, the first neuron in our chain, and they're going to project all the way back to the medulla oblongata. We'll talk about the specific area here in a second, right? But they're going to project all the way back to the medulla oblongata, synapse onto the secondary neuron, And then that secondary neuron is going to travel and project all the way to the thalamus. We know the rest of the story there. Remember, our thalamus is our relay center. And so from the thalamus, then we'll have our tertiary neuron go to the primary gustatory cortex, which is located in our invisible lobe, in the insula, the only lobe of the cerebrum that you cannot visibly see on the outside of the brain. Okay, so it's a 3-neuron chain. Primary neurons, or the sensory neurons, come from the tongue back to the medulla oblongata. Then from the medulla oblongata, our secondary neuron projects to the thalamus, synapses onto the tertiary neuron. The tertiary neuron then goes to the primary gustatory cortex in the insula. So you might know that you have a taste, you have 5 different taste sensations. Now, if you recall, the definition of a sensation is going to be when you have conscious perception of whatever those neurons were used to monitor a stimulus. So we have 5 basic taste sensations. Now, we're all pretty familiar probably with the first 4. The 5th one might be a new one that you're probably not familiar with. You have sweet, salty, sour, and bitter. And then umami, which is going to be more of a meaty type of flavor tasting, and that's due to the amino acids. All right, so sweet is going to be produced by organic compounds, natural sweet sugars, for example, if you're eating an apple or an orange or a peach and whatnot. Salty comes from the metallic ions, sodium, potassium, iron. Sour comes from items that are acidic. If you recall, they have a low pH. Like vinegar is a great example. Lemon juice. And then bitter is going to be something that is going to be alkaloid or basic. That means they have a high pH. And if you've ever tried unsweetened chocolate, you know what I'm talking about. I worked at a restaurant many years ago, and there was somebody, one of the cooks in the back of the kitchen there, was giving people, well, not giving them, but he was betting people that he would give them $50 if they could eat an unsweetened chocolate bar in one bite. And of course, everybody said, all right, that's an easy bet. I'm gonna tell you something, I tried it, I couldn't do it. Unsweetened chocolate is not what you think. And then finally, we've got our umami, and that's going to give us that meaty flavor, right? And that's because of the amino acids that are present there. So when we're talking about how we are going to convert that stimulus energy, right? That's that chemical energy that's involved in those tastings. Remember, we have to convert that chemical energy into nerve signals. And that process is transduction. So how do we transduce, all right, whatever the taste sensation is into a nerve signal? Well, when we're dealing with sweet, bitter, and umami tastings, we are going to utilize the secondary messenger system. That means that these tastings are going to bind onto the receptors, all right, on our gustatory cells, and they're going to trigger the activation of the G protein. The G protein is a protein on the inside of the cell. That in turn will then stimulate an enzyme, all right, that will activate the opening of cation channels. All right, we saw before in the olfaction, our G protein would activate adenylate cyclase. Adenylate cyclase, which is the enzyme, would convert ATP into what we called cyclic AMP, and then that cyclic AMP would run around and open up these cation gates. And then that would allow sodium to enter into the cell, calcium to enter into the cell. But what it does is it results in depolarization. And that's what we're looking here. All right, so we're going to be using, all right, our secondary messenger system, utilizing the G protein, and that is then going to cause depolarization. Now, it's a little bit different when we're talking about salty and sour tastings. because these folks are based on ions. Well, that's nice, right? So we don't even need to worry about utilizing the secondary messenger system with salt and sour tastants, because these tastants, since they're ions already, they'll directly depolarize the cell. So we don't have to deal with that secondary messenger system. But ideally, that's what we're trying to do. We're going to try to depolarize, right, these sensory cells to generate our action potential. And when that occurs, then that neurotransmitter, all right, is going to be released from our gustatory cells onto the sensory neurons, all right, that reside in cranial nerve 7 and 9. And they're going to then transmit that action potential back towards the brain. And that's how we look at that. So here's how we do it. Okay, quick review. Our primary neurons, which are going to be the sensory neurons of cranial nerve 7 and cranial nerve 9, they are going to send that sensory information back to the medulla oblongata, specifically to the nucleus solitarius. Now from here, a couple of things can happen. We'll have some other neurons that are located in the nucleus solitarius, and so those will get triggered, and they will start to go to other digestive centers in your brainstem. And that will start your mouth to salivate. That's part of the digestion process. And usually when you are tasting food or liquid, well, that stuff that's in your mouth is eventually going to wind up in your stomach. So we got to turn the stomach on. We've got to start releasing digestive enzymes in the stomach. We need to start to waken up the smooth muscle cells in the stomach walls so we can start mixing all of that stuff in your stomach up with the digestive enzymes there. So we're going to activate stomach secretions. Now, unfortunately, if you do taste something that you do not find pleasant, all right, something that's nauseating, you can trigger the gag or vomiting reflex, right? And those reflex centers are also located in the brainstem there. So it's all close by. So the nucleus solitarius sends out, all right, neurons to these other areas, right? But what will happen is that primary neuron synapses on the secondary neuron, and that secondary neuron will travel from the nucleus solitarius up to the thalamus. And then it synapses onto the tertiary neuron. That tertiary neuron will then go to the primary gustatory cortex in the insula. And then that's where you actually have the conscious perception of taste. In addition to that, you will also get information on how hot or cold, whatever it is that you're tasting, what the texture of whatever it is that you're tasting. I've talked to many, many people, and several people do not like to eat sushi based on texture. Well, guess what? That information is going to be transmitted, right, from those sensory neurons in the mouth and in the tongue all the way back here to the primary gustatory cortex here. And we'll also integrate some of our sense of smell. And that's how there is a connection between gustation and olfaction. That's how, when you have a cold, you'll notice things, you can't taste things as much or as well, because olfaction plays a role in that, because there is a bit of integration in with olfaction. All righty. There's the gustatory pathway. Let's take a peek at that. Put it in picture form. Here's our friend bringing that cup to their face to drink down whatever the liquid is, right? At the same time, they're getting some odorant production here so they can smell whatever it is that they're drinking. So the tastants enter into the oral cavity, right? And they'll stimulate, okay, the neurons here, either in the anterior portion of the tongue or the posterior portion of the tongue. Depending on what area is going to determine if it's cranial nerve 7, which is on the anterior portion, or cranial nerve 9, which is on the posterior portion. And that will then stimulate those sensory neurons, they'll transmit that sensory information here to the medulla oblongata, to the nucleus solitarius. And then from there, all right, the secondary neuron will then travel up to the thalamus, synapse onto the tertiary neurons, and then the tertiary neurons will then wind up here, right, in our gustatory cortex in the insula. And then you get that conscious perception of the taste. So that is the gustatory pathway in a nutshell. Okay, onward to vision. And we're going to start off by talking about the photoreceptors, the visual receptors that are located in the retina of your eye. So when we're talking about our photoreceptors, there are three things that the photoreceptors are able to detect. The first thing is the light intensity. The second is going to be color. And the third is going to be movement. So our photoreceptors are going to provide us, all right, via whatever type of visual stimuli that we're getting on those three categories, right? Light intensity, the color of the light, and if there's movement going on. And so those photoreceptors will help to give us that visual information of whatever it is that we're looking at. And so we're going to go through and talk about that. So I want to start off by talking about phototransduction. Again, we know that transduction is converting the stimuli energy into electrical signals, action potentials, nerve signals. So in this case, when we talk about phototransduction, we need to convert that light energy into the electrical signals. And so this is going to be performed by our photoreceptors. And there's two types. We have our rods and our cones. And we'll get into the specifics of those two different types of photoreceptors. But let's just look at the general appearance of a photoreceptor. There's four parts. We have an outer segment, an inner segment, cell body, and the synaptic terminals here. So when we're looking at the outer segment, this segment of a photoreceptor is going to be embedded in what we call the pigmented layer of the retina. And I believe you should probably remember that from last week here. We find our picture here. Here you can see we're looking at the retina, which I could zoom in a little bit more. Right? The retina has two layers. It's got the neural layer, which is made up of all of these cells here. And then it's got the pigmented layer, which is this little spot right here. You see those brown cells there? All right, that's the pigmented layer. So the outer segment of our photoreceptors are embedded here into the pigmented layer. So we have the outer segment, then our inner segment, and that contains all the organelles of the cell, like the mitochondria, Golgi apparatus, all that fun stuff. Then we have our cell body, where the nucleus of the cell is going to be located. And then at the other end here, we have our synaptic terminals. And inside the synaptic terminals, we have our synaptic vesicles filled with our neurotransmitter. We'll talk about the neurotransmitter. It's glutamate, and we'll talk about its role here, right, in vision. Okay, so keep that in mind. The outer segment, the inner segment, the cell body, and our synaptic terminals there. Whoops. All right, so what's important about the outer segment is here is where we have these discs. And in these discs, we have this structure called the photopigment. And this structure contains, all right, the necessary machinery that is used to absorb the light that comes in through the pupil there. And we're going to talk about it. We're going to get into more detail here about what the photopigment is and all that fun stuff. Okay. Keep in mind also, right, the neurotransmitter for the synaptic terminals is glutamate. So we're learning a new neurotransmitter. So let's talk about the two different types of photoreceptors. We have our rods and our cones. All right, the rods are going to be much more abundant than the cones. All right, but the thing about rods is right? They are very sensitive to light. So much so that they work very well in dim light. So this is like your night vision. So when you wake up in the middle of the night, maybe to go to the bathroom or have a bowl of ice cream, if you don't turn the lights on and say there's a little bit of light coming in from the moon or whatever, right, the rods are going to be activated and you're going to be relying on them, all right, to see. Problem is, all right, the rods, all right, aren't very good with visual sharpness. They provide more of a blurry image. And that's true. And so when you're wandering around in the middle of the night, you're not going to be able to see the fine details of maybe some of the pictures on the wall in your house as you're going downstairs to have that ice cream or whatnot. All right. But what we will see, all right, is that the rods are going to be found more on the periphery of the retina, whereas the cones are going to be more centrally located. And we'll talk about that in a moment. But what we're going to see here with the rods is, all right, you'll have several rods that are going to converge on the next layer of cells in the neural layer called the bipolar cells. And then those bipolar cells will converge and synapse onto the last layer of cells in the neural layer, and that's called the ganglion cells. So this is not going to be a very good picture here, but bear with me. All right, so these are a bunch of these rods, okay, your photoreceptor cells. And so all these rods will converge and onto a couple of our bipolar cells. Then our bipolar cells will converge onto the next group of cells, which are our ganglion cells. So what we're seeing is what we call that neuronal convergence there, that's similar to what we saw when we were talking about the sympathetic nervous system there, kind of a similar concept. So you're going to have like 100 rods that are going to converge onto, like, say, 50 bipolar cells, and those 50 bipolar cells will converge onto like 25 ganglion cells. All right. Because of this makeup, we're not going to have a very good image. The image sharpness will be compromised because of that. But what we will see, if you want like a crisp, clean image, then we want to use the other type of photoreceptor cells, which are the cones. Now, the problem with cones is they don't work very well in dim light. They work better when we have more light. I don't want to say high intensity light, but like daylight. All right. In addition to that, all right, they work best when there's more light available. But in addition to that, they also give us our color vision. Easy to remember. Cones begin with a C. Color begins with a C. Okay. So it's our cones that give us that color vision. And the nice thing about our cones is it has a 1-to-1 relationship with the bipolar cells and the ganglion cells. So we're not going to have a blurry image. We're going to get a nice sharp image. But of course, we need bright light for that. We don't have ample light, like the light, if you were to, I mean, I'm not lecturing to you in the dark in my room, but because I have the light on, I have, my cones are providing me with sharp images of whatever's in my room. But if I were to turn the light off, right, and look around the room, I'd be relying mainly on the rods, and they're not going to give me a very good image. It'll be blurry because of all that convergence that's going on. All right, so here you can kind of see in our outer layer here, those are those discs that I was telling you about that are stacked on. Now, these discs are constantly turning over. Okay, so the older ones will get phagocytized, all right, by the cells there in the pigmented layer, and we'll just add on some new discs. But if we look close here in our periphery of our disc, we're going to see, all right, this molecule, this structure here called the photopigment. And the photopigment, all right, is made up of an opsin protein and then retinol, all right, which is the structural component here. We're going to talk about it in a second that helps with our vision. Retinol comes from vitamin A. That's why when I was growing up, my mom would say, always eat your carrots because they'll help with your vision. Right? But what she didn't know is, yes, that's true, all right, but it's specifically the vitamin A in those carrots because it's in high amounts that will help my vision. So let's talk about these photopigments. Again, these are going to be, all right, the structures that when light strikes your retina, all right, these structures are going to absorb that light that's coming in. Because remember, we got to convert that light energy into electrical energy. We got to get it into an action potential. All right, so here we're looking at our photopigments made up of our opsin protein and then the actual molecule that absorbs the light, the retinol. And what we'll see is, all right, our rods have a specific type of photopigment, and that's called rhodopsin. And then our cones, they have their own photopigment, and that's called photopsin. But here's the spin with the cones, all right? The cones have 3 types of photopsin, and it's based upon, all right, one of 3 colors, either blue, green, or red. Notice that those are the primary colors of the spectrum there, blue, green, and red. And so one type of cone will have, will be able to detect blue light. Another type of cone will be able to detect green light, and then another type of cone will detect red light. Only one color can be detected by a cone. It can't detect both blue and red, right? And that's based on the type of photopsin that is going to be present, right? So blue cones are going to detect short wavelengths because that's what blue light travels on, a shorter wavelength, whereas red cones are going to detect a larger wavelength, a longer wavelength. And then green will be somewhere in between. Okay, so rods have rhodopsin and cones have photopsin, but there's 3 different types of photopsin. Okay, blue cones have one specific type of photopsin, green cones have another, and then red cones have another one. All right, so keep in mind, all right, that's what these pigments are going to, or these structures are going to be able to do. They're going to transduce, all right, the type of color based on the wavelength of that light. So if you look here at this picture here, you'll notice, all right, blue cones have a significantly shorter wavelength, all right, it's 420 nanometers compared to green cones and red cones. And so it really depends on, all right, what type of photopigment is present in that photoreceptor will determine the color when we're dealing with our cones. That brings us to our first clinical view today, which is color blindness. It is an X-linked recessive condition, which means we'll see it in males more often, all right, than females, because as you know, all right, males have an XY chromosome, whereas females have an XX. So in order for something, all right, to knock, well, I shouldn't say knock out, but in order for you to be colorblind, you have to knock out, all right, the X chromosome. Well, males only have 1 X chromosome. Females have 2, so it's harder to knock both of them out. That's why we see it more often in males. Okay, so the colors that are most often affected are red and green. My father-in-law is colorblind, and he likes to hunt. So the problem is, all right, if he wounds an animal and it runs away, most hunters usually follow the blood trail, and they find the prey, and then they, you know, finish it. All right, but he unfortunately is unable to do that. But from what he's told me, he's never missed. So he's never had to follow a blood trail because he's that good. And so he says. So let's talk about phototransduction and how we are going to convert, all right, that light energy into an electrical signal. And so we're going to start off by talking about what happens in our rods. Okay, so when we're dealing with the rods here, all right, our rhodopsin, that's our photopigment, right, contains the opsin protein and then our retinal component. But in the dark, the retinal is in a specific type of configuration. All right, so let me just show you in picture, and then I'll come back and show you in the words. Okay, so here's our photopigment molecule, okay, rhodopsin. Here's the opsin protein, and it has the retinal, all right, component here attached to it. And it's in what we call the cis configuration, which means we have a slight bend here, all right, in our hydrocarbon. So what happens is in the dark, no problem, all right? Our cis retinal is attached to the opsin protein, all right? We're in a dim light, so this photopigment here is fully operational. Now, what happens is you flip on the light, let's say to get up to go to the bathroom, and you flip on the light, forgetting that, you know, the light is going to blind you momentarily. And so what happens is the light rays come in and they change the configuration of our retinal protein from the cis configuration to where it's bent here, and it straightens it out. And so it transforms it into the transretinal configuration. So here's the problem with that. Because of that, we've changed the configuration of that molecule. The transretinal, okay, is going to disassociate from the opsin protein. It just pops off and it leaves. Okay, it wanders out, all right, of the disc, and it travels into the pigmented layer of your retina. So this molecule pops right off the protein and it travels into the pigmented layer of the retina. And it's there that we need ATP to bend that molecule back into the cis retinal configuration. So it makes sense, right? Like if you want to try to bend a bar, okay, or bend something, you got to use some energy to do it. So the ATP is going to then be utilized to change the transretinal molecule back into the cisretinal configuration here. And when that happens, then that cisretinal can come back onto the opsin protein here, and then we can start utilizing that again, only in the dark though, right? Because those light rays are going to inactivate that. So that's what happens here, right? When we're talking about our phototransduction, we're going to reconfigure that cisretinal, right? Once light hits it, it changes and it causes it to straighten out into that transretinal configuration. And we call this the bleaching reaction. So the problem is, because of the bleaching reaction, all right, we have to reconstruct, right, that molecule. Because remember what I said, the retinal has disassociated off of the opsin protein. So we've got to put it back together to get that photopigment to work again, right? So this is what happens, why the rods don't function in bright light, because of this bleaching reaction, right? So in the dark, they work great, all right? But in the light, not so much because of the bleaching here. And so what will happen is you go to the bathroom, you've done your business, you turn the light off, you're back in your dark room and you lay there. Try it tonight sometime, right? You'll probably notice that you can't see as well, all right, in the dark as you initially were able to see when you first got up. And that's because we need to regenerate the rhodopsin photopigment. And so it takes roughly about 20 minutes to 30 minutes, all right, for our photopsin to regenerate. So that's what brings me into dark adaption and light adaption. Right, so when we're talking about the bleached rods, right, after the bleaching there, takes about 20 to 30 minutes. But when we talk about light adaption, all right, where our the cones are affected, right, like for example, walking out of a movie theater in the middle of the day into the bright lights, right, what we'll see is our cones, all right, don't take as long. Right, they'll only take about 5 to 10 minutes. And that's because the photopsin there in the cones regenerates faster than the rhodopsin does in the rods. Also, we'll get some pupillary constriction here, right, which is good because when the pupils constrict, it lets less light into the eye. Our rods are more on the periphery, so this kind of saves the rods. But unfortunately, the cones are located right there in the fovea centralis. Right? There's going to be some rods in there too, but the majority of our cones are found in the fovea centralis, and that will blast the cones first, sparing the rods because, again, like I said, they're more on the periphery of the retina there. All right, so let's talk about now how we are going to convert that light energy. We talked about how it stimulates the photoreceptors. Now let's talk about how it gets converted into action potentials, into our nerve signals. Right? So we'll start talking about the rods first. Okay, the rods, right? When we're in the dark, keep in mind, in the dark, your night vision is going on. Right? What we're doing here is we are depolarizing our rods. How do we do that? Well, you have this molecule called cyclic guanosine monophosphate. And so what this cyclic guanosine monophosphate, or CGMP, its job, think of it like a doorstop, right? Like, you know, those wedges that people will jam under a door to keep it open. I think of cyclic GMP as this wedge. And what it does is it binds onto the cation channels in the membrane of the cell, and it keeps them open. Well, if these channels are open, it's going to allow sodium and calcium to enter into the cell. We call that the dark current. And the dark current flows into the cell, and it depolarizes that cell. So that depolarization will lead to the opening of the voltage-gated calcium channels in the far end of our cell at the synaptic terminals. And so it causes those channels to open up and stay open as long as a dark current is being produced. And that causes our neurotransmitter to be released, which is glutamate. So our photoreceptors are going to release their neurotransmitter onto the next cell, right, which is the bipolar cell. But unfortunately, the glutamate hyperpolarizes the bipolar cell. And because of that hyperpolarization of the bipolar cell, it causes, all right, the ganglion cells not to be excited because the bipolar cells are hyperpolarized and they cannot stimulate or excite the ganglion cells. So that means the ganglion cells are not going to produce any nerve signals. So let me show you that picture first, then we'll talk about what happens in the light. So this is what we're seeing. All right, so here we are in the dark, okay? That's when the rods work great. They love the night vision here. All right, so we're seeing this cyclic GMP, right, keeping open our cation channels, constantly being produced in the dark. So because it's being produced, it keeps these channels open. Our dark current, our calcium and sodium flow in, causing depolarization. That depolarization will trigger the opening of the voltage-gated channels, and those voltage-gated channels then will stimulate the release of the neurotransmitter glutamate. That glutamate is released upon the bipolar cells, and it hyperpolarizes them. That means the bipolar cell, its voltage-gated calcium channels stay closed. They don't open up. And so it does not release its neurotransmitter, and that means we do not excite the ganglion cell. Okay, so that brings us to what happens in the light. All right, let's talk about that. So here's what happens. Once there is light, ample light, right, we are going to see hyperpolarization of the rods. How does that happen? Well, we already talked about the bleaching effect. That's what I'm talking about when I say the light splits the rhodopsin. All right, so we get the bleaching effect going on, but also we're going to bust out our secondary messenger system again. And we're going to deal with that G protein. So the G protein now has become activated inside of our rod. And that G protein then activates an enzyme called phosphodiesterase. ASE is an enzyme. Phosphodiesterase goes over to that cyclic GMP and it breaks it down. So now your cell is taken away, all right, that door wedge. All right, so we remove the door wedge. Well, what happens when we remove that? All right, the door closes. So here's what happens. All right, those cation channels close. We're no longer going to be depolarizing the cell because sodium and calcium can't enter into the cell. All right, so in this case now, we're not depolarizing the cell. We're not making the inside of the cell more positive. Now we're making it more negative. And because of that, that causes those voltage-gated calcium channels to close. And so our photoreceptor, the rod here, is not releasing its glutamate. So it's not releasing its glutamate, which means it's no longer hyperpolarizing the bipolar cells there. So now the bipolar cells can release their neurotransmitter, which is glutamate, onto the ganglion cell. And that ganglion cell then becomes activated, and it is going to send its impulses back to the brain on the optic nerve, cranial nerve number 2. Take a look and see what that looks like in picture form right here. Okay, so we flipped our light switch on. We bleached our rhodopsin, right? Then we'll see, because of that, we've activated our secondary messenger system, and that phosphodiesterase has broken down the cyclic GMP, all right, inside of our photoreceptor. So we've essentially removed the door jamb, that wedge, it's gone, right? Our cation channels all close up. We no longer have that dark current coming in. So we're no longer depolarizing the cell. In this case, we've hyperpolarized our cell. So our voltage-gated calcium channels are no longer opening up and releasing glutamate. So since the glutamate is not being released, right, the bipolar cell is no longer hyperpolarized. It's not being inhibited anymore. So it can do its thing. And so it opens up its calcium channels and releases glutamate onto the ganglion cell, and it causes the ganglion cell to now generate action potentials, which are going to travel back towards the brain. And that's phototransduction for you folks. Okay. So when you're looking at stuff, you're basically seeing light bounce off of these objects. And so when the light bounces off these objects, it's going to enter into your eye. But a couple things are going to happen, all right, while it gets there. This picture here is showing us what refraction is. And refraction is the bending of light rays. Right? And so when the light is entering your eye, ideally what we're trying to do is we want to bend those light rays enough to where we can get them to focus on the fovea centralis. That's where your cones are. The majority of your cones are going to be located for that sharp vision. So that's our goal. We want to bend those light rays to the point where whatever you're looking at is going to have a nice, sharp, crisp image. So when you look at this picture of the spoon in the glass mug there with the water, you'll notice that it looks like the spoon looks where the spoon is present in just the air. It's in one position, but then you look below that in the water, you'll notice that the spoon is in a different position. And that is showing us what happens to light rays, all right, as they pass through different media. Okay, so remember the 3 phases of matter? You've got air, liquid, and solid. basically what this picture is showing us is that light travels differently in air, it travels differently in water or liquid, and it travels differently in through solids, right? So that's what we're going to see here, right? Certain media will bend the light more and certain media will bend it less. Obviously, when it travels through the air, it's bent less. When it travels through the liquid, it's bent more. So that's what we're going to talk about here when we're talking about refraction. So let's talk about what we call the near response. How we're going to focus when we're looking at stuff that's 20 feet or less compared to how we look at stuff that's 20 feet or more. So there's three phenomena that occur during the near response. That's when we're looking at things that are 20 feet or closer. Like if you're looking at your radio in your car, you look down at your watch, or if you're on your cell phone, typing a text message to somebody. You are undergoing the near response when you do any of those things. So there's three things that occur. You get eye convergence, accommodation of the lens, and pupillary constriction. Right, so those are the three things. So what happens, right, for convergence of the eyes? Well, right, the extrinsic muscles, which we talked about in lab class last week, right, your medial rectus muscle, primary mover, is going to pull your eyes inwards, okay, toward your nose. So the eyes will move medially. And so the reason for this is we want to get the fovea centralis, all right, to be front and center. And so this is the best way to do it. We're going to get whatever it is that we're looking at onto the fovea centralis in each of your eyes. Where you might notice sometimes, especially if you've been up for 24 hours or 48 hours or near the end of the day, right? Sometimes, right, you might notice that your vision is not as sharp, especially say, for example, you're a proofreader for a publication company. And that's all you do all day long is read, 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 read. All right. And so at the end of the day, you might notice, that you're reading, it's a little bit more blurred, all right, and it's tougher to read. You won't get that sharpness. And that's because, all right, those extrinsic eye muscles have weakened. They got tired. And so they cause that diplopia, that double vision that you'll get. So that's a good time to close whatever it is that you're looking at and take a break. All right, the next part of the near response effect is going to be the accommodation of the lens. And what we're going to see here is, right, the ciliary muscle, which is located in the ciliary body there, right, in the eye, the ciliary muscle will contract. Now, because of the ciliary muscles contracting, it causes a slackening of the suspensory ligament. The suspensory ligaments attach the ciliary muscle, all right, to the lens. So when your eye undergoes accommodation, those muscles contract. It causes a slackening of the suspensory ligaments, and that allows the lens to thicken and become more rounded, give it more of a convex appearance. So it goes from something that's like this to something that's like this. Okay, it becomes thicker there. And what that does is that increases the amount of that bending of the light, gives us the ability to focus more. So we get more light refraction. Because ideally, again, we're always trying to get that image of whatever we're looking at right there on the fovea centralis. So this is another way that we're able to do that. And then finally, right, we get pupillary constriction. So what happens is the sphincter pupillae muscle, that's that muscle that sits there in the iris, right? It's going to contract. It's going to make the size of the pupil smaller. And the reason why is because that lets less light in, right? But more so, it focuses whatever light that's coming through on the center of the lens. And that keeps the image from being projected on the periphery of the retina. Because again, on the periphery are where our rods are. Well, they're not going to give us a very sharp image. They're going to be more of a blurry image. They're great for dim light situations, okay? We want our cones to be the ones that are going to be affected by this. So when we constrict the pupil, it lets less light hit the periphery of the retina, and more light is going to enter in towards the fovea centralis there. So our goal is to avoid that blurriness that we would see. All right, so now what happens when we look at something further away? Okay, so if you're driving along and you're following a car and you're like, well, I want to remain, 50 yards back from this car. I want to have a safe following distance. So you're looking at that car that's in the distance. All right, so here's what happens. All right, in this scenario, you're not going to undergo convergence of the eyes because that object is way far away. So that's going to keep, all right, your eyes are going to be forward and not medially converging. Now, what will happen is, all right, the ciliary muscles are going to relax in this scenario. When they relax, they pull the suspensory ligaments tight, and that causes the lens to flatten out. We refer to that as the resting position. So now we've gone from our accommodation, this big fat lens. Now, all right, we've thinned it out a little bit, like that. And then we see that the pupil now becomes more dilated. So the dilator pupil muscle, pupillae, excuse me, that's that muscle that sits in the iris. That's going to contract, and that is going to cause dilation, and that it will let more light in, okay? And it will, because what happened here is we have that flattened lens, right? We can let more light in because you can see we have a greater surface area here for whatever it is that we're looking at. That whole surface area there compared to this surface area here, which is smaller during the accommodated lens. So that's what we're looking at here. Let's zoom in down here at the bottom real fast. So here is our accommodation, right? So both the eyes move medially, right? You'll notice here, right, the pupil is smaller here compared to this picture, right? The pupil is dilated here, the pupil is constricted here. But you'll notice the ciliary muscles have contracted, causing a slackening here of the suspensory ligaments that allows the lens to become thicker. So whatever light that is entering is going to be hitting the center of our lens so we can get a larger amount of refraction going on. Whereas up here, all right, when you're looking at something far away, you'll notice, compare the shape of the lens. We zoom out so you can compare, you can see how it's fatter here and it's thinner here, right? So the ciliary muscles have relaxed, and so that causes the suspensory ligaments to become taut. That causes the lens to flatten out, all right? But we have our pupillary dilator muscle has, our dilator pupillary muscle, excuse me, has dilated, and so we can let more light in, hit that center of that lens, and we can get that image focused on that fovea centralis there. All right, so our second clinical view of the night is cataracts. All right, our cataracts, as we age, you'll start to develop these small opacities. Oh, if something's opaque, it's very difficult for light to travel through it, if at all. And so you'll start to get these small little opacities within the lens. And so obviously it will make it much tougher, right, for light to travel through the lens. And so you'll have a harder time being able to see things that are close by or close up with sharpness of vision. So usually what will happen is we can remove, there's some techniques in which they can use sound waves, all right, that phacoemulsification, in which they just use those sound waves to kind of break up the plaques. I know that in some scenarios they have been using lasers, okay? So we're finding all sorts of good uses for lasers. And in some cases, some folks have had to do a lens transplant, and that also can help with dealing with the cataracts there. Something that we normally see in older individuals, though, for the cataracts. All right, I'm going to stop here. I know it's a little bit earlier, but I feel like this is a good place to stop. We'll finish up the lens or the vision on our next class next week. But let's do this. Let's take